Hello, students. This is Fazan Mirza. We are discussing gene expression today. Gene expression case can be discussed at the level of prokaryotes as well as at the level of eukaryotes. The example that we are discussing in prokaryote basically deals with lac operon. Lac operon is present in bacteria, for example, in E. coli. The bacteria is having lac operon. It is a cluster of three genes: lac Z, lac Y, lac A. Lac Z codes for beta galactosidase enzyme. Lac Y codes for beta galactosidase permease protein. Black A codes for beta galactosidase transacetylase enzyme, and they all are genes. Beta galactosidase gene is LAC-Z, beta galactosidase permease gene is LAC-Y, and beta galactosidase transacetylase gene is LAC-A. The cluster of these three genes together with the operator which is present before these and the promoter for structural genes, this make up the lac operon. Now, what is the lac operon? Lac operon is actually having the genes which, when transcribed, they will give the ability to the bacteria to start to digest lactose. You know, bacteria will not have lactose excess every time, so it will not make unnecessary enzymes and unnecessary proteins in the absence of lactose. So if lactose is not available, lac Z, lac Y, lac A, these three will stay inhibited. They will not turn into mRNA. Their mRNA will not be made. Transcription will not occur for lac operon, and these genes will not be translated into polypeptides either. So as a result, the structural genes will be prevented to show themselves in the phenotype of that bacteria since lactose is absent. So for a gene to transcribe, how gene transcription occurs or gene expression occurs, for any gene to get transcribed, that gene or that set of genes will have a promoter region. This is the promoter region present. It precedes the actual genes. Here, in this case, these three genes share a common promoter. We don't have in eukaryotes, there is no common promoter of multiple genes. In eukaryotes, there is one promoter for one gene, but lac operon is an example of a prokaryote in which multiple genes share a common promoter. So what is a promoter region? Promoter region is present as a switch on a gene. So when the promoter region allow RNA polymerase enzyme to attach to it. So when RNA polymerase enzyme attaches to the promoter region, the genes that are present after that, they turn into mRNA, their transcription occurs, and because of their transcription, mRNA is made, and then mRNA can be turned into a polypeptide during a process of translation. Now, as I told you, in the absence of lactose, this bacteria will not make unnecessary enzymes, because otherwise it will only be a loss of energy. Energy will be lost in making these polypeptides, these enzymes, plus the amino acids will become a limiting factor. It might not have enough amino acids to make those necessary proteins that it need if it ends up wasting amino acids here. So in the absence of lactose, when lactose is not available to the bacteria, for example, the bacteria is growing for example, in glucose or in some monosaccharides, glucose or fructose or galactose, it will not use these enzymes for digesting those, for metabolizing those. So what will happen? Just before the lac operon, there is a regulatory gene and the promoter of the regulatory gene. The regulatory gene produces a protein called as the lac repressor protein. The lac repressor protein attaches to the operator region of the lac operon. Operator region is present between the promoter for the structural gene and the structural gene themselves. So the operator region ends up being occupied by the lac repressor protein. Who made this lac repressor protein? The regulatory gene made this lac repressor protein. The regulatory gene had its own promoter. So the lac repressor protein attaches to the operator region. In the in this attached condition of lac repressor and the operator, RNA polymerase cannot bind to the promoter of the structural gene. So as a result, the transcription of lac Z, lac Y, lac A is prevented. The lac operon is switched off. Now, let's suppose you give lactose to the bacteria. Lactose behaves as an inducer. Lactose will go and it will bind to the lac repressor protein. Lac repressor protein, once attached with the lactose, its shape changes and it detaches from the operator site. So when it detaches from the operator site, the promoter is free and exposed to attach to the RNA polymerase. So RNA polymerase comes and sits on the structural promoter, the structural genes promoter, and the structural genes start to undergo transcription. Beta galactosidase gene, lac Z, LAC-Y, beta-galactosidase permease gene, and LAC-A, beta-galactosidase transacetylase gene, they all start to undergo transcription, and then translation is followed, which ends up making the proteins of these. So beta-galactosidase is the actual enzyme that the bacteria needs to metabolize lactose. Same goes for beta-galactosidase transacetylase. Beta-galactosidase permease increase the permeability of lactose into the bacterial cell membrane or through the bacterial cell membrane. Here, there are two more terms that you should keep in mind: inducible enzyme and the repressible enzyme. Inducible enzyme is the enzyme that is synthesized only in the presence of inducer. So the enzyme beta galactosidase and transacetylase, they both are the enzymes which are inducible enzymes, and these are being made only in the presence of the inducer, and the inducer was lactose. Repressible enzymes are also the same. In this case, the repressible enzymes are those whose synthesis is blocked by a binding of repressor protein at the operator region, which you saw that lac repressor protein, when it is attached to the operator region, the RNA polymerase cannot find its attachment site on the structural gene promoter. In eukaryotes, the gene's control is a bit different. RNA polymerase alone cannot carry out the gene transcription. Apart from RNA polymerase attaching to the site of promoter, transcription factors must attach as well. Otherwise, gene in eukaryotes cannot be transcribed. So gene control or gene expression in eukaryotes is a bit more complicated as compared to prokaryotes. So here we are taking an example of how gibberellin controls growth. You know, gibberellin controls growth. It causes elongation of the plant by cell division. It also promotes seed germination. So how growth is controlled by gibberellin? Gibberellin promotes the DNA transcription. The DNA transcribes and makes certain proteins which are used in growth of the plant. So how the growth is controlled? So there is a transcription factor PIF. If PIF is attached to the promoter region here, the genes will be transcribed and the and growth will be promoted. But there is a protein called DELA protein. DELA protein is a protein that inhibits PIF. When DELA is attached to PIF, it prevents PIF from attaching to the promoter site on the DNA. So here comes the role of gibberellin. What gibberellin would do: whenever gibberellin is present, it will attach to gibberellin receptor and an enzyme. The complex of gibberellin, the gibberellin receptor and the enzyme, the complex will cause the destruction of DELA proteins. Once the DELA protein have been destructed, PIF will be free to bind to the promoter region. It will go and bind to the promoter region, and RNA polymerase will be attached there as well. And the DNA will be transcribed, and those genes will be turned on, which actually are required for the growth of this plant. Transcription factors are required in various parts of life of an organism. They are required for appropriate gene concern to be switched on. They are required for genes to be switched on in a proper sequence. Transcription factors are required for determination of sex in mammals. Transcription factors also are required for changes in the for responding to changed stimulus from the environment, for example, high environmental temperature, as well as it's the turning on of the proto-oncogenes or the tumor suppressor genes, for example, P53, BRCA1, BRCA2, which regulate the cell cycle and apoptosis and program cell death. So program the transcription factors are involved in those as well. Hormones also have their effects, or they exert their effects using transcription factors only. You may recall how the collecting ducts become permeable to water in the presence of aquaporin, which is attached to the apical surface of the membrane, because the basolateral membrane of the collecting duct is attached with the ADH hormone. So when ADH hormone attaches to the basolateral surface of the collecting duct cells, it triggers a series of events inside the cell, which initiates the transcription of the gene that codes for aquaporin. And here transcription factors are involved. So in the presence of those transcription factors, they are activated, and the gene that makes the aquaporin is transcribed, and aquaporin is made, and the aquaporin vesicles fuse with the apical surface, and the collecting duct cells becomes permeable to the to water, and water starts to move into the blood. So these are there are various examples of how transcription factors regulate various parts of life of a particular organism. That's it. Thank you so much.